So, hello everyone. So, from last class, if you guys have any queries, please let me know. Okay. And in during this session, if you guys have any, doubts or something, you can write it in chat or you can even speak if you can. So, for today, we are going to start the second part of the same chapter, chapter 9, that is biotechnology, principle and processes. In last class, we have seen what is genetic engineering, what is bioprocess, and how this modern biotechnology has you know evolve this field so much. Today we are going to talk about the tools of biotechnology. Okay, so I have switched on my camera today and I will close it in after some time because of better streaming. So if you guys have any query, any problem in during this session, please write it down in chat box, or if you feel to speak, please speak it on. Okay, so yeah, so let's start today's class. Disclaimer: what are going to be the learning objectives of today's class? First, we will understand what is the role of these restriction enzymes that we have even talked in previous class in DNA cutting, and how they are specific, how this works so specific that they cut the DNA at a specific site. Okay. Then second learning objective would be we will identify the different types of cloning vectors. I have told you in last class also what is vector. A vector is a biological molecule that transfer our gene of interest into the host organism. But today we will talk about vectors only, but we will see the another side of this vector also, that along with transferring of the gene, how it helps even in the cloning, that is to make multiple identical copies of the gene of interest. Third, we will analyze and see the selection methods for transformed cells. Okay, this is the index of today's class, which we will today's class we will follow. First, tools of recombinant DNA technology, that is 9.2 session of chapter 9 of biology NCERT class 12th, in which there are two subsection, that is 9.2.1 restriction enzymes, and second the cloning vectors, the delivery vehicles, 9.2.2. Then we will summarize our whole whatever we will study it in today's slide. We are going to summarize it and tell you about its key features. Then there will be some NCERT based questions. Then there will be a slide on keynotes, and last there will be some interesting questions that you guys gonna solve yourself. So let's start. First question comes into our mind is, what is restriction enzyme and how it has been discovered? So the restriction enzymes are discovered in 1963. So what happened? Some scientist has observed that the growth of bacteriophage in bacteria have suddenly stopped. You guys know what is bacteriophage? Let me tell you. Bacteriophage is the viruses that infect the bacteria. Okay, and what it do? It inject its genome into the bacteria and start replicating it. Okay, and start replicating its own genome. So we scientists saw that the growth of this bacteriophage genome is suddenly it's like stopped in bacteria. So the question arises, why it happens? What is the reason? So they saw that there are some enzymes that making this possible. So they have isolated two enzymes from that restricted bacteriophage growth in E. coli, and found that enzyme 1 add methyl groups to DNA, whereas enzyme 2 cut the DNA at a specific position. That specific position we got to know later, but they have seen that the second enzyme cut the DNA, and later they term it as restriction endonucleases. After this, they got to know that there is some enzyme which is responsible for cutting the DNA. They have isolated the first restriction endonucleases, that is HIN2, in 1968. They found that this endonuclease has some, you know, important characteristics. First, it always cut DNA at a particular point. They cut the DNA at a very specific point, and they find out that they recognize that these endonuclease, restriction endonuclease, they recognize a certain specific recognition sequence that is made up of 6 nucleotides, and that region is the region where they cut they cut the DNA. Okay, after finding this, that this and the restriction endonucleases work like this, that they first specific they recognize a specific recognition sequence. Okay, and that could be like from 4 to 8 nucleotide. For HIN6, HIN2, it was 6 nuclear base pairs, but for others it could be like anything from 4 to 8. They bind to it and they cut the DNA at the specific site. Then what is the current information on this enzyme is that over 900, more than 900 restriction enzymes have been isolated from more than 230 bacterial stains. So I want to ask here, like, did you guys understood like how restriction enzymes work? If you have any query, please feel free to ask either in chat box or in you come if you want to speak, you can speak. And if there won't be any question, I will think that you guys understood and I will move ahead. Okay, so I'm moving ahead. Yeah. Okay, so more than, as I told, like more than over 900 restriction enzymes been isolated from 230 bacterial strains. And what is the key feature of all those restriction enzyme? That each and every restriction enzyme recognize a different recognition sequence. Okay, this is very specific about this restriction enzyme, that they can recognize this recognition sequence, but every restriction enzyme can recognize its particular sequence. It's not like that 3 restriction enzyme can recognize a single sequence recognition sequence. No, everyone has their own unique recognition sequence. This restriction enzyme, they belong to a class of enzymes called nucleases. As the name suggests, nucleases, its function is to cleave the nucleic acid. Now, this restriction enzymes are divided into 2 types. Okay, first is exonucleases. As the name suggests, X, you can relate it to some extreme, like extreme end or something. So this is a nucleases that remove the nucleotides from the end of the DNA. Second example, exonuclease 3. Whereas the second type is called endonucleases. It makes cut at it cuts, as in it cleaves the DNA at a very specific position. Example, EcoR1. So now what does this mean, EcoR1? There is a specific nomenclature for this restriction enzymes that we're gonna talk in next slide. So yeah, so EcoR1, the E stands for, the first letter E comes from the genus, that is E from Escherichia. Okay, and the second 2 letters come from species of those prokaryotic cells from which they are isolated, that is coli. That is why we write E and CO. The fourth letter indicates the strain from which they have taken. In this case, the E. coli strain is RY13, that's why we write EcoR. Whereas the 4, that is the Roman number, that we write the Roman number 1. It indicates the order in which these enzymes were isolated from the strain of bacteria. Like this, for example, there is a bacteria, take the example of E. coli only, and the first restriction enzyme which they have taken out from this is E. coli, this restriction enzyme only. That's why it is called EcoR1. E, E stands for genus, CO stands for species, R stands from strain, and 1 is the order in which these enzymes were isolated from the strain of bacteria. I hope it's clear. If there is any doubt, you guys can ask in chat box or you can speak, okay? Is this clear from till now? So I'm going ahead. It's again the example say EcoR1 comes from E, that is Escherichia, CO, that is coli, RY13 is the strain, whereas first is the order in which this enzyme has been isolated. For example, I will give you one more example. Okay, so in we have seen that first isolated restriction enzymes were HIN2. So what is HIN2? H is Haemophilus, the genus is Haemophilus. N is influenza, the species. D is the RD, it was the strain. And second is the order in which this restriction enzyme has been isolated. Okay, so this is the order. Like this is the manner, this is a nomenclature how we write the name of, how we name this restriction enzymes. Now, what is the mechanism of action? How it works? As I told, first it recognizes the sequence. It recognizes the specific DNA sequence. This 4 to 8 nucleotide will recognize, and then it going to bind to it. This palindromic nucleotide sequence we are going to talk later, but first you understand that what it do, it first inspect the DNA sequence. When it will find that okay, this is my recognition sequence, this is where I have to cut, it will bind to that DNA sequence, then it will cut the sequence. Okay, now what is palindrome? So normally, not don't think about only biology. Normally what it is a palindrome, a sequence, or even what Malayalam. Okay, you can see that Malayalam can be written same from front and back. Like forward Malayalam spells same, and backward it also spells same. That is also the case in palindromes in DNA sequence in biology, that a sequence of base pair that reads the same on two strands when the orientation of reading is kept the same. You will understand more when I will tell you the example. Yeah. So this is the recognition sequence for EcoR1. Okay. So when you will read this sequence from 5 dash to 3 dash, it's here should be a 1 dash. Okay, so from 5 dash to 3 dash, you will read it as G A A T T C. When you will read the same to the from the second strand, but in the same direction, that is 5 dash to 3 dash, you will read it as G A A T T C. Same happens with when you will read it in the form direction from 3 dash to 5 dash. C T T A A G, and from 3 dash to 5 dash in second strand, C T T A A G. If you guys have seen, it reads same from both the direction. Okay, so this is what you call palindrome sequence. So this is the recognition sequence where EcoR1 cuts the DNA. Yeah. So what is the cutting action? So now, okay, I told you like it recognize the sequence, it go and bind to the DNA, but now how it cuts? So what happen in a palindromic sequence, usually what happens, it cuts both the strand of the DNA helix at same site, but between the same two bases. For example, I'll show you from last slide only. Yeah, so see, What happens? E. coli my arrow is visible now to you guys. Yeah, it's visible. So what happened? E. coli eco R1 comes, it recognizes the pattern, it binds here, and it cuts this strand between these spaces G and A. It will cut here between G and A. And if it is cutting between G and A in first strand, it is going to cut between the same base pairs only in the second strand also. It means it will cut it here between GA and it will cut it here between GA. Okay. Is it clear? Is it clear to everyone? Okay, if you guys are not writing anything, I think it is clear. So yeah, the first one says that it cuts both the strand of the. Okay guys, so I'm switching off the camera because there's some streaming problem. That's why the reason I'm switching off the camera. Okay, so if you have any question or anything, you should ask me in this chat box, or you can even write. Okay. So yeah. So its cutting action is that it cuts both the strand of the DNA helix at the same sites, but between the same two bases a little away from the center of the palindromic sequence sites that we have seen in the example I have shown you guys. Okay. Now what happens when it will cut the DNA, cut the space pair between G and A, it will leave a single stranded hanging stretches, and these stretches are called sticky ends. And these sticky ends going to join together with the help of the enzyme that is DNA ligase. And in this slides we are going to see the picture where you will get more better idea like how the sticky ends look and all. Okay. So as I have told that palindrome is a group of letters that form the same words when read both forward and backward. Example Malayalam, as I have told, given the example already. See, now you can understand it much better. So this is the figure 9.1. This has been taken from NCERT Biology, NCERT only. So see what happens in this. This is a vector DNA, okay, and this is the foreign DNA. These two DNA has to come together to form the recombinant DNA. So in this vector DNA there is a sequence which is EcoR1 sequence only, that is GAATTC, and same in the foreign DNA there is a sequence GAATTC. Okay, so there is one thing that you guys need to understand and remember, that same restriction enzyme should be used for vector DNA as well as foreign DNA to ligate it. Okay, you can't use different restriction enzymes for these two different things. Okay, for vector DNA as well as foreign DNA, you have to use the same restriction enzyme. So see what happened. E. coli core R1 restriction enzyme came. It cuts the DNA between the base pair of G and A here both, and when it will cut it, it will make it like this sticky end where it is act as a overhang stretching. Okay, and same happens with it when it will cut it at the position between G and A, it will make the sticky ends like this. And now this sticky ends join with the help of DNA ligase to make a recombinant DNA. Okay, you guys understood? Any confusion? Okay, so I'm moving ahead. It tells the same thing only, that a sequence of base pair that reads the same on both the strands when the orientation of reading is kept the same is called the DNA palindrome, and many of the restriction enzymes recognize palindrome sequences. For example, EcoR1, it recognizes this sequence. It comes and cut it between the base pair G and A, make a sticky ends, and then ligate it using DNA ligase. Now next. This is the NCERT question 7, or you can like see it here. I have already told you in earlier slides also that 5 dash to 3 dash, if you will read, it will be same like GAATTC, and if you read it in the that same direction from 5 dash to 3 dash, it will read same, that is GAATTC. And this is the same figures in which we have shown that what are these different steps in the formation of recombinant DNA by the action of restriction endonucleases. So restriction enzyme, I told you many of the restriction enzyme has recognized the specific palindromic sequence in DNA. It binds to it and then cleaves both the DNA at the same position. In this eco R1 case, it was between G and A. The cuts are made slightly off center, means it's not directly, cuts are not made in the center but little away from the center, and it creates the sticky ends, what you also call cohesive ends. It is the overhanging stretches. This overhang stretches comes and join together with the help of DNA ligase, and thus a recombinant DNA forms. Till now, if you guys have any question, please ask, or if you have guys have any confusion, please write it in chat box, or you can ask. Okay, so I'm moving ahead thinking that you guys understood the concept. Now, we have cut the DNA. Okay, we got a gene of interest. We got the gene which we wanted, the DNA fragment, DNA sequence which we wanted. But now how we can separate and visualize as that this is our DNA fragment that I want, that we wanted. So for this, we have a technique called agarose gel electrophoresis. So what happens in this agarose gel electrophoresis? Okay, there is no figure. Okay, sorry. So first of all, we know that DNA are negatively charged molecules. Why it is negatively charged molecules? Because DNA has this phosphate group. Okay, that's why it is negatively charged. Now, because we know that it is negatively charged, we separate them to, we separate them to move towards the positive electrode, that is anode, through an agarose gel matrix. I will explain it more, you will get to know it better in the next slide when you will see the diagram. So here is a one trick you can learn, that we know that negative ions are anions. So anions move towards anode, A and A, because anode is the positive electrode. Remember it, okay? And DNA are negatively charged, that we all know because it has phosphatase group. So what is the mechanism on the basis of which it separates? First, as I told that this is negatively charged, that's why it moves towards the anode, that is the positive electrode. But this agarose gel has this sieving effect. Sieving effect as in, it helps to separate these DNA fragments based on their size. That's why smaller fragments move farther through these gel pores than the larger fragments. Now, as a typical agarose gel electrophoresis, so what happens? See, this is the well where we put this DNA, okay, and this is the ladder, okay, and what happens? This DNA fragments move further on the side of the positive electrode, that is anode, based on their size. Smaller size fragments move fast, whereas as you can see here, the bigger and the large fragments, it will take time to move ahead. So this is how this agarose gel electrophoresis work. Because of the sieving effect, this size exclusion, size based separation, we can use electrophoresis to separate these DNA fragments. You guys have any doubts still here? Okay. Now we have separated the DNA fragments, but we can't see DNA fragments from our normal naked eyes and even in the visible region. We can't see it. So for that, we use staining method and this exposure to UV radiation. We stain this DNA with ethidium bromide and expose it to UV radiation, and we found that this DNA fragments appear as bright orange color bands in this agarose gel. See, as it is shown in the figure. So we use by using this electrophoresis, we have separated the DNA fragments based on their size. Now, using this staining method, ethidium bromide, and then expose it to the UV radiation, we can make this DNA fragments appear as bright orange color bands. After, now we can even now visualize this. So now we can separate this DNA bands. We can cut this DNA branch from the agarose gel. Okay, and then DNA is extracted from this gel piece, and this whole procedure, when the DNA is extracted from the gel piece, is called elution. Elution of this DNA from the gel piece. Now, the purified DNA fragments are ready for use in constructing the DNA by joining them with cloning vectors. Now, as we have got our DNA fragment that we wanted, now we can join it to the cloning vectors to make the recombinant DNA. So, till now, if you guys have any confusion, ask, because we have completed this session of restriction enzymes, that how restriction enzymes is used to cut our specific DNA and take it out. So now I'm moving ahead. You guys don't have any questions, so I'm moving ahead thinking that you guys understood everything. Okay. So now, as we got our gene of interest, we got our DNA. Now we need something to transfer it to host organism. So for that, we will need cloning vectors, a DNA molecule that can carry a foreign DNA segment and not only carry but replicate it inside the host cell. What are the common vectors known? Are plasmids and bacteriophages are the some common vectors that are known. So now, what are the some essential features of this cloning vector? What are the features that make it a suitable cloning vector? First, the origin of replication. This we have talked about in a little in last class also, that it is a sequence where replication starts. And any DNA linked to it will be replicated. It also controls the copy number of the linked DNA. What is copy number? Copy number is the number of the times that DNA will be present in the genome. If there will be more copy number of our gene of interest, it will be good. And if it will be less, it will be not that good. Okay. So, origin of replication is responsible for this copy number also. High copy number vectors are preferred, as I told, for obtaining many copies of the target gene, because as many as we can have our target gene, it will be good for us. Okay, so that's why we usually choose a cloning vector whose ori can give us more copy number. So is it clear that what is ori? That it is a sequence where replication starts. Any DNA that we have, the foreign DNA that we needed, it should be linked to it. That then only it can replicate it. It controls the copy number, that is the number of the times this DNA will be present in the genome of this link. And the high copy number vectors are preferred because it helps in obtaining many copies of the target gene. Second factors, which is feature which is important, is the selectable marker. What is a selectable marker? So it is a gene that helps in identifying and eliminating the non-transformants and selectively permitting the growth of the transformants. So before understanding it, we have to understand what is transformants and what is non-transformants. So transfer, so the procedure by which this our gene of interest, that is recombinant gene, is transferred into the host and it got transformed is called transformation, and the host which got transformed is called transformants. So selectable marker is a gene which help us to identify that, yes, because obviously when we are going to insert our recombinant DNA into the host, it is not possible that every host is going to, going to take it and going to replicate it. There will be some this this there will be some false all the host also that wouldn't able to take that gene of recombinant DNA. So how we can identify that, okay, our our recombinant DNA got into this host. So this selectable marker help us to identify and then also eliminate. If that host is not been transformed, it can also eliminate it and thus help us to select the transformants, that is the transformed host cell, and that is the reason it is called selectable marker. What are the common markers? Genes encoding resistance to antibiotics like ampicillin, chloramphenicol, tetracycline or kanamycin. These are the common markers. What are its okay, so how it helps? E. coli, which we are talking like for so long, that E. coli everything. E. coli when act as is usually in this case we example which we are going to talk later, usually E. coli used as host, and E. coli has this capacity that it do not carry resistance against these antibiotic. Okay. So cloning site, recognition site. Also, one of the factors of features of cloning vector should be that the vector must have few, preferably a single recognition site. Why it should be there? Because if there will be more restriction recognition site, then there will be more generation of these fragments. Then this will going to complicate the situation. If we have two recognition site very closely to each other, then the restriction enzyme will go to each of it, okay, and they'll cut it into very simple smaller fragments, which is not good for us. It will complicate this gene cloning thing. And ligation of foreign DNA is carried out at a restriction site present in one of the two antibiotic resistance gene. Okay, it's again the same thing that I have told, but with the picture. So I will explain it till quick, that this is the cloning vector PBR322. Okay, and this is a artificial cloning vector. Okay, this will you talk in the keynotes. Here you can see these are the different recognition site for different restriction enzymes. This is for BAM-H1, SAL1, HIN3, CLA1, E. coli R1. Okay, and this region is also tetracycline resistance, and this gene is also ampicillin resistance. This is the ori, that is the origin of replication. The same thing I have told, that the same pictures I have already told, that this region is for tetracycline resistance and this region is for ampicillin resistance. And I have told that what is selectable markers, it helps in to identify the transformants, and E. coli lacks natural resistance to these antibiotics. Now vector should have, again as I have already told, that vector should have minimum restriction enzyme recognition site, or we can you should prefer that only single site, so multiple recognition site can not create the multiple fragments. So now here again this topic, this we have to understand little, you guys have to understand it little more, because this topic is important. Now, how we select using this antibiotic resistance? I told you that foreign DNA can be ligated at restriction site with an antibiotic resistance gene. So this foreign DNA can go and ligate to the at the site where there is antibiotic resistance genes. Yeah, so understand from this figure. So for example, we will take our foreign DNA and we go and ligate it to the region where this is tetracycline resistant. Okay, so now because it is tetracycline resistant and we have now added we have inserted foreign DNA, so now this vector will not be tetracycline resistant. Okay, so now when we will go and plate it into the nutrient with we will go and plate it to the nutrient where it is amplicin antibiotic is present, both the recombinant and non-recombinant will grow because both non-recombinant and recombinant have this ampicillin resistance site. Okay, but now when we will take this and replica this plate on the plate nutrient media with tetracycline resistance, our recombinant will not grow. Why it won't grow? It won't grow because we know that our foreign DNA has been inserted in between this tetracycline resistance gene thing, and now it is not resistant to tetracycline, and this is how we can identify our transformed host, and this thing is called insertional inactivation. Okay, so we have inserted the DNA at BAM H1 site in tetracycline resistance gene, and because of this, what happened? Recombinants lose tetracycline resistance but retain the ampicillin resistance, and non-recombinants, they are maintaining both the resistance. So it will grow, both the recombinant and combinant will grow in media with ampicillin, but the recombinant will not grow in the media with tetracycline. Same thing has been written here, that plate transformants on ampicillin media transfer when the plate we transform when we have plated our transformants on ampicillin media, what happened? Both have grow properly, but when we have transferred this growing colonies to tetracycline media, recombinants hasn't grow. Okay, so this is the identification that recombinants grow in ampicillin, not on tetracycline, and non-recombinants grow on both antibiotics. Again, the same figure, I'll explain it once again because it is an important topic, that when foreign DNA go and bind in this BAM H1 site, which is also tetracycline resistance, the recombinant has lost this tetracycline resistance thing, but the non-recombinant has this tetracycline resistance thing. And this cloning vector also has this both the cloning vector have this ampicillin resistance thing also. So when we have grown it on the with ampicillin antibiotics, the recombinant and non-recombinant both grows, but when we have replica this plate on the tetracycline thing, only recombinant grow, and recombinant didn't grow, only non-recombinant grow. Okay, so now we can like this plating system is very tedious task. We have to use two plate, one with the tetracycline, one with the ampicillin. So there is one better alternate selection, that is the blue white screening. So what happens, usually we take this cloning vector with beta galactosidase enzyme, and this beta galactosidase enzyme has the power to convert some chromogenic substrate, that is XCAL, into blue color. Okay, so as it says, it uses a chromogenic substrate to detect color change. Recombinant DNA is inserted into beta galactosidase gene, disrupt this enzyme function, which is called insertional inactivation. And here we can see this recombinant DNA, which is inserted in beta galactosidase, will not able to convert that substrate into blue color, though all those white colors are the recombinant, whereas the blue color, which hasn't been transformed, is the non-recombinant. As I told, blue colonies are non-recombinant and white colonies are the recombinant, because in non-recombinant there is intact beta galactosidase. It is functional enzyme which is able to convert the chromogenic substrate into blue color, but in recombinants, because the beta galactosidase activity has already been disrupted, it is not able to convert into blue color. The same figure has been shown here. What is the advantages of this chromogenic method? That it is more convenient than dual antibiotic screening. It requires only single plating. It is visual identified based on the colony color. So after this, there are also some vectors can be used for plants and animals. For plants, we use a vector which is the modified TI plasmid, which obtained from Agrobacterium tumefaciens. We have seen that this TI plasmid transfer a tDNA which can cause tumor in the plants. So now what we can do it, we can modify it, make it non-pathogenic and delivers the gene of interest into the plants. Okay. Same for animals, we have seen when we this retrovirus is responsible of making a normal gene into cancerous gene, but it can make that transfers the normal gene into the normal cells into the cancerous cells. But if we disarm this retrovirus and utilize it to deliver desired gene also in animal cells also. Okay, so what is the summary of this whole thing is that it cuts the DNA at the specific sequence called recognition site. It creates sticky or blunt ends. Sticky ends are the overhanging stretch. But what are blunt ends? So what happens when there is a cut directly in between at the same position of both the strands? It will make a cut which is we call blunt end. This cut will make end which we call blunt ends. Named after bacterial species they are isolated from. Okay, it is very essential for genetic engineering. And talking about cloning vectors, it carry foreign DNA into host cells. It includes plasmid, bacteriophage, and artificial chromosome. It contains multiple cloning sites. It enables DNA replication and expression. So first question is that what is the restriction digestion and how it is useful in biotechnology? So restriction digestion uses restriction enzymes to cut DNA at specific sequence. Its application includes DNA fingerprinting, genetic mapping, cloning, construction of recombinant DNA, analysis of DNA sequences. Each restriction enzymes recognize and cut specific DNA sequences, and it can be used in different ways. Now the question asks, describe the different types of cloning vectors commonly used in genetic engineering. Plasmid, it is small, circular DNA, double standard DNA. It gives high copy number. It is easy to manipulate. Its size limit is approx 10 kilo base pairs. And 1 kilo base pair means 1000 base pairs. So you just can think. Example, PBr322. It is an artificial plasmid. Okay, if you will ask, so this PBr322 is like P stands for plasmid 32. BR is the name of the scientist who developed this. I think it was Bolivar and Rodriguez. And 322 and 322 is the name that these guys have given to this plasmid to differentiate it from the other plasmid which they have isolated in their lab. Then this bacteriophage viral vectors can carry larger DNA fragments because they can, example lambda-phage, better transformation efficiency they shows. The other is called cosmid. It is hybrid of plasmid and phage, lambda-phage, and it can carry upon 35 to 45 kilo base pairs DNA. It combines advantages of both systems. Then its back, that is bacterial artificial chromosome. It can also carry a very large DNA fragments, that is up to 300 kilo base pairs. It has low copy number because as the DNA fragment size is large, more metabolic pressure will be on this host. It is stable in bacteria. Then this yak, that is yeast artificial chromosome. It has the largest capacity, that is 1000 kilo base pair, and it can propagate it in yeast cell. What are the keynotes that you guys should take is from here that the restriction enzyme was first discovered in bacteria by Werner Huber, Hamilton Smith, and Daniel Nathans. Over 3000 restriction enzymes have been discovered. ECOR R1, HINT 3, and HINT 2 are the first three restriction enzyme that has been isolated and characterized. PPR322 was the one of the first artificial cloning vector, and this can be used to clone DNA fragments up to the 10 kilobase pair plus in the case of plasmid and 50 kilobase pair in the case of cosmid. Yak, that is artificial yeast artificial chromosome, they can carry up to 1000 kilobase pairs, and it typically includes plasmid, bacteriophage, cosmid, BAC, and GACs. These are the interesting questions that you guys can go and solve on your own. And if you guys have any question, please write down in chat. Thank you. Let's solve this TPP today for today. So yeah. So what first question says? The first question says, which enzyme is responsible for cutting DNA at specific recognition sequence? As we have been studying it for so time that the enzyme which is responsible for cutting DNA at specific recognition sequence is restriction endonucleases. Save and next. Second question says, what is the purpose of the origin of replication in a cloning vector? So let's see the option. First, to identify transformed cell? No. It's done by selective markers. Second, to control the copy number of linked DNA? Yeah. We can control the copy number of linked DNA with the help of, origin of replication, that is ORI. To provide antibiotic resistance and to cut DNA at specific site? No, it's not the function of cloning vector. Save and next. In gel electrophoresis, why do DNA fragments move towards anode? So as I told that anode is a positive electrode and DNA is a negatively charged molecule, so it is going to move towards it. But let's see the options. First option says that DNA is positively charged. No. Second says that agarose gel has a negative charge. No, there is no charge for agarose gel. It's the sieving effect which is helping in this gel electrophoresis. Third, DNA is a negatively charged. Yeah, this is the answer. Fourth, ethidium bromides impart a charge. No, we all know that ethidium bromide is for staining purpose. Fourth question says, which feature helps differentiate recombinant DNA colonies using a chromogenic substrate? So antibiotic resistance? No. Chromogenic substrate means substrate which can induce some color difference or something. Insertional inactivation of beta galactosidase? Yes, because we know that if the beta galactosidase, what a beta galactosidase can do, it can convert a chromogenic substrate, okay, and turn it into a color. So as we have studied in our lecture that it can convert the substrate into blue color, but if there has been insertional inactivation of beta galactosidase, then it won't be possible. So this answer is second. How are bacterial cells made competent to take up the recombinant DNA? So this question, this part is something that we are going to discuss in upcoming classes. So, but the answer will be that we can use by treating it with calcium chloride. This treatment happens, and so answer is treating with calcium ions. But don't worry, this question is not from today's lecture. But still, we will save it. We saved it actually, save, and we will submit it. And we will see how many questions we have done right. No change will be allowed after submission. Yes, we will give Submit. See, all of our questions are correct and we scored 20 out of 20. So from today class, oh, very good, Hariram. Second C, you said second C. Let me see. No, it's not C because this origin of replication is not providing any antibiotic resistance. Okay, it's B because it's helping us to control the copy number of linked DNA. If you have any doubt, you don't feeling like to speak in speaker, you from mic you can A2, okay. 2A, no, it's not even to identify this transform cell. Origin of this transform cell we can identify using selective markers that we have talked in the class. Origin of replication is responsible for the starting of the replication of the DNA and to control the copy number of linked DNA. This transform cell we can identify through the help of selective marker. So I am stopping the lecture today. Thank you.